Right, good morning everyone. As you probably noticed, I'm still nurturing a little bit of a cold, so if I cough sometimes in between, my apology beforehand. I'll try to touch as little as possible here as not to spread it to the rest of the consortium. So today I'll be giving an update on our project, the transcriptomic atlases of the craniofacial sutures. And as you all probably know, the vertebrate skull, in the vertebral skull, the bones are separated by fibrous joints called sutures. And at the very basic level, these sutures are broken up in the osteogenic front, which is found on either side of the suture, and separated by the suture mesenchyme. The osteogenic front is where differentiation of pre-osteoblast to osteoblast occurs, leading to the deposition of new bone and growth of the suture. And the suture mesenchyme really separates the two. So the example that I've shown here is for a calvario suture, but the morphology of these sutures also changes between the different craniofacial sutures. There can be end-to-end overlap, and there can be differences in the width of the suture and the extent in which these osteogenic fronts interact with each other. So differential gene expression and patterning of gene expression really underlies suture development. And the two canonical examples that I've shown here are FGFR2 and TWIST1. So FGFR2 is primarily expressed in the osteogenic fronts, whereas TWIST1 is expressed in the mesenchyme, but also to a lesser extent in the osteogenic fronts. Gain of function mutations in FGFR2 lead to Apert syndrome, whereas loss of function mutations in TWIST1 lead to Satterthwaite-Johnson syndrome. And both of these syndromes are characterized by premature suture fusion. And interestingly, for both of those syndromes, we also have mouse models that really recapitulate the phenotype that we see in humans very well. Of course, these are just two of the genes, and we know that many other genes are going to be involved in suture development. So what we set out to do several years ago was to do a very comprehensive profiling of craniofacial sutures. So we've selected 11 different sutures that vary in location, in cell lineage, in bone identity on both sides of the suture, and in structure, so either end-to-end overlapping or more distant from each other. And we are profiling these for both wild-type mice and the mouse models that I indicated before. So the way we are going about this is doing laser capture microdissection of frozen tissue slices. And as indicated here, for each of these sutures, we cut out the suture mesenchyme and also the osteogenic fronts on either side of the suture. For homologous bones, we basically pool together the osteogenic fronts on either side of the suture and profile them as one. For non-homologous bones, we treat the osteogenic front separately and we profile them separately. So an overview of the wild-type sutures expression profiles that we have been generated is shown here. So every row here indicates a different suture. Every column here indicates a different biological replicate of each of the 3 different time points that we are profiling. So for the coronal suture, we take E14.5, and then for all other sutures, including the coronal suture, we are profiling E16.5 and E18.5. And then the different symbols in this chart basically indicate whether we are profiling a pool of the osteogenic front or whether we are profiling the individual slices of the osteogenic front separately. Then for the mouse mutant model, so the FGFR2 and TWIST mutants, for Apert, so the FGFR2 mutant, we are essentially recapitulating the same profiling that we're doing for the wild-type sutures. And for TWIST1, we are profiling the affected frontal and coronal sutures. So to date, we have produced datasets for 7 different sutures, which are shown here. For each of these different suture types, we have complete datasets available. And as you can see, for each of these sutures, the main differentiating component of them is a separation between the osteogenic front and the suture mesenchyme. But within each of these sutures, we also see expression profile differences between wild-type versus mutants, and also developmental time point differences. And what is really important to realize here that overall, across all of these different sutures, we've actually profiled more than 7,000 differentially expressed genes that are either differentially expressed between suture regions, across development or between mutant and wild-type mice. So there's a lot of expression data to profile and to process, and the way that we are going about this is by performing co-expression network analysis. And in these co-expression network analysis, we're basically identifying genes that are consistently co-regulated with each other across different suture regions or across time points. And we then use these correlations in gene expression profiles to build a correlation co-expression network as the one that I'm indicating here for the wild-type frontal suture mesenchyme. And each of the nodes in this network corresponds to a gene, and the connections between these genes indicate the degree by which they are co-regulated across the conditions that we are interested in. So, of course, these networks get very big. It's very hard to study them as a whole, so the approach that we're using, the Magena co-expression network analysis, allows us to take this bigger network and divide it into submodules that are increasingly high resolution. So we're basically starting at the root, which contains the entire gene network, and then we progressively break this up into smaller and smaller network modules that are hierarchically linked to each other. So in the next slide, I'm basically showing a different view of the same network. So now instead of having the nodes represent individual genes, we actually have the nodes represent individual modules within this larger network hierarchy. And in the center of this network, we have the root, so that consists of the entire network. And then as we're sort of progressively breaking down this network in smaller modules, you can see based on the top-level Go annotations that we have annotated here for each of these modules, we can start to differentiate different pathways that we know to be involved in suture development. And that includes FGFR signaling pathways, Wnt signaling pathways, and cell-cell adhesion pathways. So to take a deeper dive into our co-expression network analysis, we are also building these kind of matrices. So here, each column indicates a different module in the network that I was just showing you, and each row either corresponds to differences in gene expression profile. So these can either be differences in gene expression between the osteogenic front or the mesenchyme, or they can be differences in time or differences between mouse mutants, and that's shown here in the rows in green. And then in blue, we basically annotate the same modules for different gene ontology terms, and that allows us to sort of differentiate different pathways within these modules. And then the one module that I highlighted here in red in the column is a particular module that is consisting of genes that are more highly expressed at E16.5 in the frontal suture mesenchyme compared to later time points. And these genes encode proteins that are involved in extracellular space or cell-cell junction contacts. So if we zoom into this module, and now we're basically looking at an individual module, and the nodes in this module correspond to genes again, and the connections between these genes indicate the degree by which they are co-regulated. We can start to sort of build new biological pathways or extend known biological pathways. As you can see, most of the genes that I'm highlighting in this module have increased gene expression at E16.5 in the frontal suture, and sort of center in this module is CDH1, which encodes for E-cadherin. This is a very important cell-cell communication protein that is also that sort of regulates cell-cell adhesion, but also is involved in sort of signaling between cells. As you can see here, this particular gene is indicated by a triangle, and that means that this gene is more connected to other genes in this network module than would be expected by chance, and that indicates that this is a likely regulatory protein in this particular module. And based on what we know in the biology, that makes really perfect sense for what we're seeing here. Of course, there's more going on than just changing gene expression. Genes can be upregulated, genes can be downregulated, but we also know that genes express different transcript isoforms. So in addition to just looking at gene expression differences within suture regions or over time, development time, or between mouse mutants, we are also looking at differential gene expression. And the events that we're interested in are sort of shown here in the top left: exon skipping, mutually exclusive exons, alternative 3' and 5' splice sites for known exons, alternative promoter usage, and alternative 3' exon usage. And in the example that I'm showing here for the coronal suture, the vast majority of events that we're detecting are exon skipping events, but we also see a fair number of alternative promoter usage, alternative 3' and 5' splice site usage, and to a lesser extent, alternative 3' end usage. And if you look in the top right, if we look at the alternative splicing patterns, either between the osteogenic front and the suture mesenchyme at different time points, we see that at each development time, we see fairly similar profiles when it comes to splicing differences between the front and the mesenchyme. And to a lesser extent, and because we're here looking at the coronal suture where we have both the parietal bone and the frontal bone, we also see some differences between the splicing in the parietal versus the frontal bone as well. Overall, though, most of the differences that we're detecting are really between the mesenchyme and the front, and we haven't found any significant splicing differences between different developmental time points. So just to highlight some of the examples that we're finding, I'm showing here 3 different genes, so periostin, PLOT2, and RUNX2. Periostin is an extracellular matrix protein, and we see basically increased skipping of exon 17 in the osteogenic front. For PLOT2, we also see increased exon skipping, and for RUNX2, we also see increased exon skipping towards the 3' end. So RUNX2 has been previously shown to express different transcript isoforms that play a role in development. The different isoforms that have been described mainly differed in the 5' end, so using alternative promoters, but we're seeing in our data set that there are also other differences more towards the 3' end. And in all these cases, these exon skipping events affect domains that are central to the function of these proteins. So they are likely to alter the way these proteins functions in the suture, and that sort of adds an additional layer of complexity to our understanding of regulation within suture development. Of course, all the data that I've shown you so far is derived from bulk RNA-Seq data from each of the suture subregions. And that said, we know that there are differences in cell population even within these suture subregions. For example, in the osteogenic front, as I previously said, there is differentiation ongoing from pre-osteoblast to osteoblast. And we can basically take a closer look at this if we look at sort of staining patterns of gene expression. So in the examples I've shown here, we have RUNX2 and BCL11B. So RUNX2 is primarily expressed in the osteogenic front, whereas BCL11B is primarily expressed in the mesenchyme. But you see at the intersection between those 2 layers, there is also co-expression of these genes in certain cell types. So we know there is increased complexity within these suture subregions that we are not really capturing as well with our laser capture microdissection data, which is why we applied for and received a supplement to our main project that allowed us to look at single cell sequencing of these different sutures and basically do a more specific cell type and lineage profiling of some of the same sutures that we're also profiling in our main project. So we selected, in this case, 4 wild-type sutures: coronal, lambdoid, frontal, and sagittal. And out of these, to date, we have profiled 3 of them, and of 2 of these, the coronal and the lambdoid, I'll be showing some data today. So we're profiling these at E18.5, so this is similar to the time points that we've used in our main atlas, but also at adult time points, P10 and P28. The approach that we're using here is chromium single-cell sequencing, and the basic principle here is we use microfluidics to sort our cells. So each cell eventually gets separated into a unique droplet together with a bead. That bead contains unique barcodes, and then within that droplet, we essentially lyse the cells, do a cDNA reaction, and the cDNA reaction, because the barcodes on each of these beads differs, allows us to uniquely tag the RNA-Seq data coming from an individual cell. At the end of the day, all of these different cDNA libraries get pulled together and they are sequenced in one sequencing run. But by then analyzing and separating the barcodes out at the end of the day, we're able to reconstruct the individual expression profiles of individual cells. Now, it's important to understand that while this gives us cell resolution in our data, it typically gives us less data to work with. So in the bulk RNA-Seq data, we're generating, let's say, 30 to 50 million paired end reads for each of the different subregions. If we look at our single cell data, we're getting between 40 and 100,000 reads per cell. So we get several orders of magnitude less data, but the data that we're getting is from individual cells, and that allows us to do some very interesting things. And one of the things that, of course, is of primary interest is clustering the different types of cells that we're seeing in the individual sutures. So the examples that I'm showing here is the P10 lambdoid suture, where in the plot on the left, which is a t-SNE plot, every dot represents an individual cell that we've profiled with single cell sequencing, and they're clustered and colored according to the cell types that we have assigned based on the presence of expression markers that we know are associated with certain cell types. So in this case, we see that in red and in yellow, the osteoblast and mesenchyme really separate out very well from other cell lineages, which are mainly chondrocytes and hematopoietic cell lineages. And this really sort of allows us to really do a deep dive within each of the sutures and drill down in the specific cell populations. And what you can see on the right is that each of these clusters is associated with the expression of certain types of marker genes that are really very cell type specific for each of these individual clusters. And this allows us to generate new hypotheses regarding the expression patterns of those genes and how they're involved in regulating suture development. So we can take the data from an individual suture, but we can also do a cross-suture and cross-region comparison. And what I've shown here, the left two bars are basically the breakdown of the individual cell types in the coronal sutures, whereas the rightmost two bars are the breakdowns of the different cell types in the lambdoid suture. And then for each of these sutures, we're showing on the left E18.5 compared to P10, the adult stage on the right-hand side. And as you can see in this plot, the main differentiating factor between the coronal, the coronal and the lambdoid suture is the presence of a large population of cartilage-derived cells. So this is mainly due to the fact that the cartilage is more difficult to separate in the lambdoid suture compared to the coronal suture. But even if we discount the cell population of cartilage cells, you can still see that there are temporal and suture differences in terms of the specific cell populations that we're seeing. And this is both in the extent and the relative distribution of suture, mesenchyme, and osteoblast, but also in the extent of the presence of other cell lineages within these sutures. Focusing now on just the mesenchyme and osteoblast, as we know that these are sort of lineage related to each other, we can do what is called a lineage analysis, and you can sort of consult with Anna and Anna's poster that she's presenting on this later today for more details. But the basic principle here is that we are ordering now our different cells in the mesenchyme and osteoblast according to their pseudotime lineage. So we're basically trying to rank the expression, and we try to rank the profiles of these cells in a way so that we can reconstruct the developmental lineage of these cells. And that's highlighted in the plot here on the left, where we show basically the progression from mesenchyme to osteoblast according to pseudotime. And what you can see in the plot in the top right is that this progression is really characterized by the expression of different types of genes that mark mesenchymal genes or osteoblast genes. And you can see that these patterns change over time during differentiation. Also shown here in the top right, in the bottom right, where basically each of these different cell subpopulations that we identify within the differentiation trajectory from mesenchyme to osteoblast is characterized by the expression of different genes. And this really allows us to study in vivo the progression and the differentiation of mesenchymal cells to osteoblast, and formulate new hypotheses in the ways that we can sort of approach these differentiation trajectories. Another important thing, and this is kind of going back to the point that I mentioned, that the depth of sequencing data that we're generating in our bulk RNA-Seq project is much greater than what we get at the individual cell level, but we can actually leverage the information that we get from our single cell data to further annotate the networks and the co-expression modules that we're seeing in our bulk RNA-Seq data. And just to go back to the example that I was showing in the earlier slide about the module that is related to extracellular space and cell-cell communication, we see that this particular module is highly enriched for genes that we know are expressed in one of the mesenchymal cell modules. So again, this allows us to sort of specifically formulate hypotheses that we're now validating in the lab to see how these different modules and how these different genes are involved in suture development. And with that, I really wanted to acknowledge everybody that is involved in the project. So Greg and Mimi that are mainly sort of working on the biological end of things, generating the sutures, generating the expression profiling, and doing the validation. Whereas the people in my lab, myself, Anna, Divya, are mostly involved in data processing and data analysis, with roles for Bin Zhang's group as well in terms of the co-expression and network analysis. And with that, I'd like to open it up for questions.
So we have any questions? Okay, hold on.
So since you can really compare the bulk RNA-Seq with the single cell, and you've done both for these sutures, very beautiful. So do you have an idea of what is the loss you have in terms of transcription factors, which are usually expressed in many cases at very low levels when you do the single cell RNA-Seq?
So we haven't done a formal comparison. What I would say is, when we look at our bulk RNA-Seq data, we typically, in each of the different suture subregions, we detect expression of maybe 12 to 13,000 genes. In the single-cell RNA-Seq data, it's in the several thousand, let's say 2,000 to 3,000. So there is definitely a loss in genes that we are not capturing as well in the single-cell data as we do in the bulk RNA-Seq data. But we get enough of the marker genes from the single-cell data so that we can do differentiation or further differentiation, further annotation of our bulk RNA-Seq data.
Yeah, I guess a practical question in terms of the utilities of the network analysis. One question is, are any of the modules or genes you've discovered relevant to human disease? I'm wondering what your thoughts are about the practicality of informing.
If you look at, for example, the mouse mutants that we've profiled, we do see certain modules that are enriched in genes that are differentially expressed between wild type and mutant mice. So yes, there are modules that we can sort of link to disease that way.
Anybody else? Okie doke. Next up is Joan.